Hello, and welcome to part 5 of Innate Immunology. In this lecture, we will be talking about pattern recognition receptors. These are those sensor systems in the second line of defenses found in the innate immune system. So these are actually cellular receptors. There are 3 different classes, and we're going to talk about each of the 3 classes. And these receptors are a very important part of the bridge between the innate immune system and the adaptive immune system. The pattern recognition receptors are receptors on cells that will actually recognize and detect a microorganism or a virus. They can detect molecular patterns that are unique to bacterial cells and viral genetic material. There are 2 different types of molecules, or I should say patterns that they recognize. The first are called MAMPs, sometimes referred to as PAMPs. And that would be microbe or pathogen associated molecular patterns. These are things like peptidoglycan, flagellin, particular toxins, the LPS or lipopolysaccharide found in the extracellular membrane of the, or that outer lipid layer of gram-negative microorganisms. DAMPs are damage-associated molecular patterns, and these are self-receptors or self-patterns that these receptors recognize when there has been cellular damage, usually caused by either injury or more often by infection. So here in the bottom right, we have what are called endogenous DAMPs. These DAMPs are things like necrotic cells that are releasing molecules. So these molecules here would be recognized by a cell. Injury, of course, cellular debris, and those sorts of things. Signals being sent out by a cell that is actually infected by something, an intracellular microorganism or a virus. And then exogenous DAMPs are anything that's outside of the host cell that's actually entering into it. So again, this could be a virus entry or bacteria or intracellular bacteria. MAMPs or PAMPs, microbe-associated molecular patterns, include things like, again, peptidoglycan, flagellin, molecules that are unique to bacteria or viruses or extracellular pathogenic organisms. We are already born with receptors that recognize those. So phagocytic cells, some sentinel cells, not all receptors are found in all cells, and not all cells have all receptors. So we're going to take a look at these real quick. So the 3 classes of pattern recognition receptors are toll-like receptors. Toll-like receptors are membrane-bound. They are always found embedded in a membrane, and they are found in the cellular membrane itself, or they are embedded in an endosome or phagosome. Nod-like receptors, or NLRs, are found in phagocytic cells and sentinel cells. Sentinel cells would be like a kidney cell or a heart cell, a cell that is non-phagocytic and not part of the immune system and kind of stays in one place. So TLRs, we have nod-like receptors. They are found in the cytoplasm. And the others are the RIG-like receptors, or RLRs. And the RIG-like receptors are also found in phagocytic and sentinel cells, and they are also found within the cytoplasm. All of these are protein receptors that are a key component of the innate immune system. So here are the toll-like receptors. There are 10 that are currently known in humans. It's believed there may be 2 or 3 more, but right now we know of 10. Again, they are membrane-bound and found in the cellular surface. So here's our cell membrane, and they are integral or transmembrane proteins. They will send a signal if peptidoglycan, here we have the LPS in gram-negative organisms. So TLR4 recognizes the LPS of gram-negative organisms. When that LPS, we'll make this line the LPS, when that LPS comes into contact with the TLR4, TLR4 is going to cause some type of signaling pathway to be initiated. In this case, it's called a MAP kinase cascade. So this signaling pathway will begin inside of the phagocytic cell, or inside of the cell that is coming into contact with that gram-negative bacteria. That signaling pathway will send a signal to the nucleus, right? So it goes to the nucleus. And in the nucleus, it will trigger transcription factors, like we learned in Unit 2. It will trigger transcription factors to initiate the transcription of specific genes. Those genes will cause the production of some type of cellular response. That response could be a compound, it could be a set of proteins. Whatever it is, they're usually referred to as co-stimulatory molecules, and it's going to initiate a cellular response. So in this case, if a gram-negative bacteria, right, it has its LPS, it binds to TLR4. If that same gram-negative bacteria is coming into contact with cell and it's motile and it has flagella, then flagella would bind to TLR5, which will also initiate some type of cellular signaling cascade that's going to go to the nucleus and cause the cell to begin transcribing or producing some type of cellular response. So what type of cellular response is going to occur depends on, of course, which TLR receptors are actually being bound. Now we have TLR receptors on the surface of these phagocytic cells, and if you notice, they recognize things that are bound to the outside of bacteria, like peptidoglycan, the lipoteichoic acids that are seen in that are found in gram-negative organisms. Lipoproteins, these are proteins with fat molecules attached to them that are found in the cellular membrane of bacteria. The LPS that we already talked about, flagellin on flagella. Here we have toxoplasma is actually, right, it's called profilin-like, and it's a particular toxin. This is a suspected TLR. Notice it's TLR11. In the endosome of these phagocytic cells, we find unmethylated DNA, single-stranded RNA, and double-stranded RNA. All three of these forms of genetic material are usually viral related, and unmethylated DNA is bacterial DNA. So bacteria do have methylated DNA, but in phagocytosis, those methyl groups will be removed, and unmethylated DNA is also, oh, I messed that up. That's wrong. All right, so just wanted to kind of delete that and go over this again. So I know. So the toll-like receptors. Toll-like receptors, there are 10 known in humans. They are membrane-bound receptors, and they are found on the cell surface. So they are, here you can see they are transmembrane or integral proteins that span across the cell membrane of the cell. This is the inside, of course, and you can see the outside. They are found embedded in the membranes of endosomes, phagosomes, in addition to the actual cell membrane itself. Now, the cell membrane bound, right, these TLRs here, TLRs 1 and 2, TLR 6, 4, 5, and here is 11. This is a proposed TLR over here. If you notice, all of these TLRs on the cell membrane, they all recognize components of bacteria that are found on the outside of the bacterial cells, flagellin, LPS, peptidoglycan and gram-positives, lipoteichoic acids, lipoproteins. These are all structures and molecules that are found on the surface of bacterial cells. Now, if, let's say, a gram-negative cell is present, and this phagocytic cell is going to phagocytize it, it will come into contact with that gram-negative cell, and it will recognize here the TLR4 will recognize the LPS found on the outside of the gram-negative bacterial cell. When that LPS binds to this receptor, these receptors are proteins. So when you bind something to a protein, it changes shape. That change in shape of that protein is going to initiate other proteins that are bound to it. Now, you don't need to know the names of all of these proteins or anything. I just want you to understand the general process here, because we are going to come back to this in activation of our adaptive immune response. So as this protein changes shape, this causes initiation or activation of proteins that are also attached here. These proteins then go through what's called a signaling cascade, a series of proteins that activate each other until eventually one of them is a transcription factor that gets activated in the nucleus of the cell. And that transcription factor is specific to a set of genes in the nucleus of the cell. It causes the cell to begin transcription of those genes, leads to translation, and of course cellular product. So some type of response from the cell is going to be initiated because the TLR4 specifically was bound. The same thing will happen with any of the other TLRs over here and the TLRs that are in the endosome. Same thing. Anytime a receptor is bound, it's going to initiate that cell signaling pathway. Now, toll-like receptors are membrane bound, again, in the cell membrane and in the endosome and phagosome. In the endosome or phagosome, the TLRs that are bound here recognize antigens or components, genetic components of bacteria cells and of viral cells. So we have single-stranded RNA, which would be perhaps from bacteria, a bacterial cell. We have unmethylated DNA. This is viral DNA. So if it's a DNA virus, it can be recognized after phagocytosis, or double-stranded RNA. And double-stranded RNA is also a form of RNA, viral RNA. Human cells don't produce, we don't make double-stranded RNA in that sense. So this type of RNA, again, would be viral. So these guys here, these TLRs, are recognizing after phagocytosis and the capsid's been broken apart, or the bacterial cell has been opened up and lysed open in the phagosome or in the endosome, then these TLRs will recognize it and do the exact same thing. They will send a signal to the nucleus, causing this phagocytic cell to begin releasing and producing cytokines. These cytokines are going to be signals to other cells that something is happening. They are sounding the alarm, and the cytokine-induced production that these cells will carry out once they're bound by their antigen is that alarm signal for the adaptive immune response. Now, the other two classes are the NOD-like receptors and RIG-like receptors. Both of these are internal. They are found in sentinel cells as well as in our phagocytic cells. And when we talk about the phagocytic cells, we're talking about macrophages, dendritic cells. Some TLRs are found in neutrophils, these highly phagocytic cells. But the two that are really super important for the TLRs are the dendritic cells and the macrophages, because that cellular response is going to become important when they move into the lymphatic system to activate the adaptive immune response. Now, the NOD-like receptors are cytoplasmic. RIG-like receptors are also cytoplasmic. There are approximately 23 different NLRs. I'm not sure how many RLRs there are. NOD-like receptors are focused primarily on intracellular bacteria infections. So if we look over here at the NLRs, they will recognize bacterial secretion. They recognize, here we have an NLR for flagellin. Here's another one that might recognize cellular damage caused by intracellular bacteria infections. In antigen-presenting cells, they trigger a very specific response, and this is called the creation of an inflammasome. And the inflammasome is an inflammation initiation complex. So they initiate an inflammation. RIG-like receptors are also cytoplasmic proteins, but instead of recognizing intracellular obligate bacteria, they recognize viruses. So again, they start seeing uncapped single-stranded RNA. Remember, eukaryotic RNA, our own RNA, has what's called a 5' cap. If the 5' cap is missing, then a RIG-like receptor will recognize it as viral RNA. Recognizes double-stranded RNA, another form of viral RNA as well. We do see RLRs in almost all cells. It is the early warning system for a viral infection, and they are responsible for initiating production of what are called AVPs, or antiviral proteins, as well as the interferon response, and they also cause cells to produce interferons. Now, in AVPs, antiviral proteins, these are small little proteins that the cell itself will also produce, but it sends out a signal, and it sends out a chemical signal to neighboring cells in autocrine signaling or in paracrine signaling. And these will tell neighboring cells, right? So I have some neighboring cells over here. It warns neighboring cells that there's a virus on the loose, so that neighboring cells begin producing their own set of AVPs. These antiviral proteins will help prevent entry of that virus, so that virus can't get in now and cause all kinds of problems, right? So here's our little virus with its spikes. And if this virus were to try to get in and try to begin an infection stage, these AVPs here may be able to prevent that. So it not only tries to save itself, which it may be too late if there's a viral infection, but it also tries to warn neighboring cells and say, hey guys, there's a virus out here. You need to prepare for that. So over here on the right, we have just kind of, this is from your textbook, and it's really just showing initiation of that interferon response and how this works. So here we have a virus-infected cell, and this is an RNA virus. It has single-stranded RNA, and when that RNA, let me get my pen back here, when that RNA when that RNA begins to replicate, it becomes double-stranded RNA. That is part of the life cycle of the virus itself. That double-stranded RNA is going to activate a set of genes responsible for interferon production. These cells then release interferon into the interstitial fluid, into that extracellular matrix, and that interferon begins diffusing to the neighboring cells. A neighboring cell will receive this interferon and begin producing those AVPs. Now, because there is no current viral infection, those antiviral proteins that are being produced by that cell are inactivated. So in this state, they're called an IAVP. Later, since this cell over here is infected, we know from the viral replication cycle that these viruses are eventually going to be released out of the cell. When they get released out of the cell and the virus tries to enter into a new cell to cause infection, we see these inactivated antiviral proteins become activated, and this will actually induce apoptosis of the cell. So it's not that it's going to necessarily allow for survival of the cell, but it's going to induce apoptosis. And with apoptosis, if this cell dies, my pen is not working for some reason here, but if the cell dies and is no longer functional, it can no longer serve as a virus factory. So the cell dies prior to viral replication, and this helps to reduce the viral load in the actual cells themselves. So just for a quick review on pattern recognition receptors, you want to know the difference between the damage-associated molecular patterns and the pathogen. I learned them as PAMPs. They have since changed it to MAMPs, microbe or pathogen-associated molecular patterns. You want to know the three classes of receptors, where they are found, what types of cells they are in, the types of things that they recognize, be able to compare and contrast TLRs, NLRs, and RLRs. And then these other questions look at their role in innate immunity and how they act as a bridge between innate and adaptive immunity. So these are some really important review questions. Make sure that you go through each of them individually. You want to make sure that you understand what's being asked, because these are important questions on the exam. Hope you enjoyed the lecture, and I'll see you guys in part 6.